Hello and welcome to Science Research Weekly, Season 4, Episode 16, Busting Tauopathies. Get ready for DNA mutation rates across generations, a potential Alzheimer's vaccine, tissue expansion mass spectrometry, updating old methods in SAS, planning a statistical analysis, and 7 new cancer grand challenges. This episode covers material from April 18th to the 24th. Let's get started with some research articles. First up, human de novo mutation rates from a 4-generation pedigree reference. Researchers assembled genomes from 4 generations, 28-member family, to look at human de novo mutation rates. They found the mutation rates vary for more than an order of magnitude depending on the genomic context. And just really with this pedigree, looking at a lot of genetics, they were able to more flesh out how the genome mutates across time and families. Next up, the article targeting of phosphorylated tau at threonine 181 by a Q beta virus-like particle vaccine is safe, highly immunogenic, and reduces disease severity in mice and rhesus macaques. So researchers tried out this vaccine on 2 different strains of mice, sort of genetically predisposed to tau accumulation, and then rhesus monkeys, so a non-human primate model, and to look at preventing the accumulation of tau proteins in a brain, which is the hallmark of things like Alzheimer's and other tauopathies. They used 2 doses and really were able to reduce pathological tau and brain atrophy while improving or at least maintaining memory and motor function. These animal trials, including the earlier trials that this research group did, really suggest a translational utility in humans. So, and I think even more so, especially with the translational aspect, they found that it was safe. That there wasn't really any side effects. All the mice and macaques got through the treatment procedure. So that's really promising results to translate into the next stage. All right, last article for today, TEMI, tissue expansion mass spectrometry imaging. Researchers were able to combine tissue expansion with laser-based mass spectrometry to profile biomolecules within the tissue while preserving their spatial distribution. And I've seen things like this before where they, like cell expansion or other expansion, where you're able to essentially get the substrate larger, and so you can more easily image all the stuff that's in it. So very interesting stuff here. All right, we'll switch over to our research tools. First up, Rick Wicklund from the Do Loop has an article on updating old SAS programs. He looks at PROC LOESS. Essentially what he does is he updates graphs using the more modern features and then compares them to the old ones. Because one of the nice things about SAS is programs that ran 20 or 40 years ago can still be run today. But he kind of shows how, well, that's still the case and that's great. You can look at how newer codes can do things more smoothly. Kind of interesting comparison. All right, over on the analysis factor, the post shares strategies for choosing and playing a statistical analysis. Very important, especially for sort of beginners or people just getting their feet wet. Questions that this post answered, how to go about this, making sure things like the tests are actually answering the research question, taking into account the design of the study, taking into account the level of measurement and distribution of the independent and dependent variables, and of course, dealing with any issues in the data. It's always helpful to think through how you're actually going to analyze measurements from some sort of experiment, and so this might be a helpful guideline. All right, no new R bloggers posts of note, but 2 nice crayon packages to unpack. First up is Circulize Plus, using ggplot2 feature to write readable R code for circular visualization. I've used, I think, just Circulize. So in earlier edition, I don't know if it's by the same group, but it may be helpful if you want to look at circular things, especially with like genomes taking prokaryotic circular genomes in particular. Anyways, I digress. The next one is DLM fit miss, fitting GLM, so that's channelized linear models, with missing data in both responses and covariates. All right, let's close with some research funding. Following up on Tauopathies, the Department of the Army from the Department of Defense has an Alzheimer's research program transforming research award for supporting non-incremental research designed to reduce risk or prevent the development of Alzheimer's disease or Alzheimer's disease-related dementias. Allowable research includes projects utilizing animal models as well as clinical research projects. So maybe that group looking at that vaccine could apply for this and do some clinical research. Fantastic. And finally, there is the Cancer Grand Challenges. There are new challenges available and currently open for what they call expressions of interest. This would also be like letters of intent or letters of interest. The 7 challenges are AI-human collaborations in cancer, cancer avoidance, the dark proteome, my personal favorite, mechanisms driving mutational signatures, the nervous system in cancer, rewiring cancer cells, and TEMI, tumor microenvironment dynamics. Tauopathy or bust. Thanks for joining me for Science Research Weekly. Science on.